Welcome, everyone. So glad to have you here for today's explainer. Think of me as your professional forensic mentor for the day. Now, if you've ever watched a police procedural on TV, you know the drill, right? Investigators perform absolute magic. They solve these incredibly complex murders in 45 minutes flat, surrounded by glowing blue lights, typing on computers that just seem to know everything. But as fun as those shows are, the real science of crime scene investigation, it's far more rigorous, it's incredibly specific, and honestly, I think it's a lot more fascinating. So today, we're taking off those Hollywood lenses and digging into the genuine scientific methods used to actually solve crimes. Right off the bat, let's talk about the real people behind the microscopes. On TV, you usually see this one lone detective interrogating suspects, dusting for prints, and then casually running a complex DNA sequence. In reality, well, forensic teams are highly specialized. If you're looking at this as a potential career path, you're not going to get there with instant television magic. Real investigators rely on hard science. You're looking at essential undergraduate degrees that are heavily focused on foundational biology and chemistry just to get your foot in the door. And hey, if you want to be the medical examiner calling the actual time of death, you'll need a full medical degree. For aspiring lab technicians, your day-to-day is going to involve running highly repetitive, rigorous tests under some pretty intense pressure. Securing an internship in a local crime lab is absolutely your best path forward to gain that crucial hands-on experience. But no matter the specific role, everyone in this field shares one core mission: using strict scientific protocols to gather and analyze evidence that will actually hold up in a court of law. Okay, let's dive into this. We're going to walk through a hypothetical training scenario today, a murder case set in Chicago. Here is our investigation roadmap. Number 1, estimating time of death. 2, digital evidence limits. 3, facial recognition. 4, blood chemistry. And finally, 5, DNA analysis. Part 1, estimating time of death, also known as the mortis phases. Let's set the scene. Our scenario begins in a freezing Chicago alleyway at 11 p.m. First things first, before touching a single thing, investigators strictly secure the scene. This establishes a tight chain of custody, ensuring any physical evidence remains legally sound for the courtroom. Inside this secured scene, they find our victim, Bob, and he's got a 5 p.m. receipt in his pocket. Let's look at the biological clock medical examiners use to narrow down exactly how long Bob has been dead. The first thing they look for is livor mortis. That's really just a Latin term for how the blood pools. You see, once the heart stops pumping, gravity takes over, and it gives the skin a purplish look. If a body has been dead for over 12 hours, that pooled blood coagulates and dries right in place. But Bob's blood? It's still liquid. This confirms what we suspected from the receipt. He's been dead for less than 12 hours. Next, they check for rigor mortis, which is the stiffening of the muscles. Here's a mind-blowing biological fact for you. Your body actually requires chemical energy to make your muscles relax, not contract. When you die, that energy supply completely stops, so the muscles just lock up. This stiffening usually starts about 2 hours after death and lasts until about the 36-hour mark. Since Bob's muscles are stiff, we know for sure he's been dead for more than 2 hours. So putting that together, Bob has been dead somewhere between 2 and 6 hours. To get more specific, the medical examiner has to take the body's core temperature. This is usually done rectally, a decidedly unglamorous detail those TV shows always conveniently skip. Bob's temperature is 29 degrees Celsius. Now, normal human body temperature is roughly 37 degrees. Under normal conditions, a body loses heat at a highly predictable rate of 1.5 degrees Celsius per hour. This biological process is called algor mortis. Since Bob lost exactly 8 degrees, the basic math suggests he's been dead for just over 5 hours. That puts the murder right around the time he left the store. All right, as your forensic mentor, it's time for a quick knowledge check. How does a freezing 5 degrees Celsius Chicago winter night affect our algor mortis math? Well, the answer is that environmental factors completely disrupt the standard formula. The freezing air literally causes the body to lose heat way faster than that standard 1.5 degrees per hour. Because it is so cold out there, our medical examiners can't pinpoint an exact time. They actually have to widen the window, placing the time of death broadly anywhere between 5 p.m. and 7 p.m. Part 2, the limits of digital evidence. With our timeline scrambled by the cold weather, investigators head over to the convenience store to check out the security cameras. On the tape, they spot a distant, highly pixelated figure stepping out from behind a tree and following Bob down the street. And this brings us to one of the biggest myths in television history, the magic of zoom and enhance. Seriously, on a TV procedural, a tech just taps a few keys, the blurry face suddenly becomes crystal clear, and boom, they've got their killer. No way. The scientific reality of digital evidence is totally different, and it all comes down to strict pixel density limitations. A digital image is basically just a color map made of tiny squares called pixels. The color of every single pixel is recorded as an exact permanent average of all the colors within that tiny physical space. And this brilliantly illustrates our next knowledge check. Why can't we just use software to enhance that blurry stalker's face? Well, it's because once a camera averages those colors together to make a single pixel, that data is permanently fixed. You literally cannot reverse engineer an average to pull out the tiny specific details that were blended together in the first place. Say a dark green pixel is averaging out a bright green logo on a black jacket. Zooming in just gives you a bigger dark green square. The data simply isn't there to be found. Part 3. Facial recognition and biometrics. Fortunately for us, the stalker actually returns to the convenience store 3 hours later, giving the security camera a nice, clear, close-up shot of his face. Now we can finally utilize computer pattern recognition. Humans are naturally great at recognizing faces, right? We do it all the time. But computers, they have to be mathematically taught how to do it. They do this by analyzing a face print. Human features are arranged in highly specific, unique ways, like the exact curvature of your eye sockets or the precise distance between your nose and your mouth. Computers measure these unique dimensions to create a biometric face print. But, and this is important, unlike TV, where detectives instantly search a flawless national database of every single citizen in seconds, those systems don't really exist. Real life usually relies on local databases. Chicago, for instance, uses a system called NeilFace that cross-references local police mugshots. In our hypothetical case, the investigators catch a really lucky break. The local database successfully matches the face print. The man in the video is Charlie, a local hardware store owner. Detectives go back to the convenience store footage. They see Charlie putting something on a shelf, and when they go inspect that exact shelf, they find a wrench with a dark red stain on it. Part 4. Blood chemistry and the Kastle-Meyer test. So, we have a dark red stain on a wrench, but in a court of law, you can't just walk in and say, hey, it looks like blood. It could be red paint, it could be rust, it could just be chemicals from his hardware store. The forensic team must prove exactly what it is, scientifically. Let's head to the lab. So, the crucial point is, unlike those magic glowing sprays you see on TV, real chemistry is sequential and it's precise. To perform what's known as the Kastle-Meyer test, lab technicians follow 3 strict steps. Step 1, swab the stain. Step 2, apply a specific chemical called phenolphthalein to the swab. Step 3, add a couple of drops of hydrogen peroxide. To understand why this actually works, we need a super quick chemistry lesson. Blood contains hemoglobin, and hemoglobin acts as a catalyst. A catalyst is simply a substance that forces a chemical reaction to happen. When you mix these specific chemicals together, the hemoglobin in the blood steps in and allows the hydrogen peroxide to physically strip the hydrogen atoms away from the phenolphthalein, essentially turning that stripped molecule into water. Let's test what we just learned. What actually causes the swab to change color? If you followed the chemistry there, it's that new, hydrogenless form of phenolphthalein that turns a vivid, bright pink. If there was no blood on that wrench, there would be no hemoglobin to act as a catalyst, the chemical reaction just wouldn't happen, and the swab wouldn't change color at all. Because our swab turns pink, investigators now have scientific proof that the stain is, for sure, blood. Part 5. DNA matching and STR analysis. All right, we know Charlie followed Bob, and we know Charlie's wrench definitely has blood on it. The final step is linking that blood directly to our victim to secure a legally sound case. We need to do a DNA analysis. DNA is the complex molecule made of long strings of base pairs that makes you entirely unique, well, unless you have an identical twin. Now, in the real world, it would take far too long and be way too expensive to sequence an entire DNA strand for every single piece of evidence. Instead, forensic biologists use a really clever technique called STR analysis, which stands for short tandem repeats. Basically, everyone's DNA has certain sections where the base pairs repeat in a pattern, but the number of times that pattern repeats is completely unique to you. By isolating and looking at just 13 of these specific repeating sections, the lab team can actually create a profile in under an hour and a half. Now, what's really interesting about this slide is the sheer statistical power of this method. It is staggering. By matching just those 13 specific sections, the odds of 2 people having the exact same base pairs in all of them are roughly 1 in a billion. That means there are probably only 6 other people on the entire planet who share your specific STR profile. For a court of law, that level of statistical certainty is absolutely accurate enough to definitively identify a suspect or a victim. The STR analysis is totally conclusive. The blood on that hardware store wrench absolutely belongs to Bob. Systematically, we've gone all the way from the crime scene to the lab to build a highly compelling case. But remember, as forensic professionals, your job isn't just running tests in a vacuum. It requires real soft skills to communicate these complex findings clearly to a jury, all while ensuring that strict chain of custody is maintained so the evidence doesn't get thrown out. Forensic science provides the facts, not the narrative. We know Bob's blood is on Charlie's wrench, but did that wrench actually inflict the fatal blow? Or was someone else entirely involved in that dark Chicago alley? That is where the science ends and the detective work truly begins. I really hope you enjoyed this deep dive into the real science of forensics. Stay curious, keep looking past those Hollywood myths, and I'll see you in the next explainer.